Good afternoon, everyone. Welcome to the series of the NCITA webinars. Today is our first webinar entitled Optimizing Imaging in the Omics Era. I'm sure you all know that NCITA stands for the National Cancer Imaging Translational Award. And really the main goal of NCITA is bringing the research together, moving from the silos of a single center to more collaborative approach where the effort is brought together to actually translate from one center to another and ultimately to the broader community. We have a series of 3 talks today. The first speaker will be Professor Eric Aboagye, starting off with challenges and opportunities for radiomic studies. Eric is a professor of cancer pharmacology and molecular imaging at the Imperial College in London. Eric, the floor is all yours.
Okay, let me share my screen. Okay. So thank you, Evis. It's a pleasure to kickstart this first set of webinars with a topic on challenges and opportunities of radiomic studies. No relevant disclosures. And what I want to do is touch on 2 things, opportunities and challenges. So radiomics is a rapidly evolving area, and together with deep learning constitutes probably the highest growth area in medical imaging. Radiomic is an imaging analysis approach that converts the image data into high-dimensional, mineable feature space. What I want to add is the ability to link this to various phenotypes and genotypes so that we can place it in the context of cancer research. So 2 of the first studies that really highlighted the potential of this area were by Segal, who showed in 2007 that they could use 28 imaging traits to reconstruct up to 78% of global gene expression. Subsequently, in 2014, Hugo Etz and Philippe Lambert, their collaborators, were able to also show that there was a strong relationship between radiomic features and gene expression. But importantly, also showed that there were very strong prognostic features for lung cancer, which also translated to head and neck cancer. I want to place this into context, and what we tend to do in imaging is we have several outputs that we want to address. So let's say, for instance, we want to address the issue of accurate diagnosis and staging. With that in mind, you also want to reduce recall rate. And what we propose in this paper is to combine different approaches and multiplex them to achieve a particular output. And the issue here is that radiomics is able to be used. We can use radiomics to multiplex several of these outputs. So 2 examples by way of opportunity, and then I'll speak to challenges. So one from our own lab, where we use an unsupervised approach to tease out prognostic vectors, and then another for the field of immunotherapy. So the workflow for most radiomic analysis are pretty simple. In our case, we use a software called TextLab version 2, but this would apply similarly to PyRadiomics and several others. So it starts off by segmenting lesions, converting these into matrices, and extracting various radiomic features, including texture, shape, size, intensity features, wavelength decomposition, and then link these to various cancer phenotypes. So in our case, we look at features from first order statistics, second order, and higher order statistics. Because we apply these wavelets, we end up with about 665 different features to work with. So this is an example of ovarian CT, and this is an area where we've done quite a lot of work. What I want to highlight is that this epitomizes convergence science. One of the good things we did at a very early stage was to bring together a team with different questions and to drive this approach. So here we've got molecular biologists, bioinformaticians, radiologists, surgeons, oncologists, and the idea is to bring this team to work on the problem. Now, if you look at this image, you can already see some texture within the image, and if you expand the image, you can see runs of pixels. You can see also within this image various clusters of intensities, and there's these types of data that allow us to run various radiomic feature extraction algorithm. This is just by placing the intensities on this. And I want to talk through our approach to this. And I think in this case, we had tried to understand the data. And so we initially just did some simple spectral clustering to try and group the data by comparing each patient's radiomic feature. And in the data clustered into three clusters. And cluster one, or group one, was the high-grade serous group. And that was correlated with copy number aberrations and also poor progression-free survival. And so once we'd done that, we decided to really delve into the high-grade group. And again, within this group, we found clusters in an unsupervised approach, where those clusters were also correlated with biological phenotypes such as ascites. There were two clusters, and one cluster obviously had poor survival with those, and those had with the group that had ascites. And then we decided to build a model. And so then we used lasso, and again, one could use various approaches. Our approach was predicated on a cutoff value of with a false discovery rate of 0.05, and then to apply lasso to that. And so we minimize, lasso allows you to minimize and select the optimal set of features, and then they independently validates that and compare that to molecular profiles and response to treatment. So we used for that proteomics, targeted RNA sequencing, and SNP arrays for the copy number. We came up with 4 features. So the vector comprised of 4 features: fractal dimension, gray level run length matrix, a neighborhood gray tone difference matrix, and a first order statistic. And so if you sum these with their weights, you end up with a vector. And that vector with the k-means clustering allowed us to define decision boundaries, and those decision boundaries were a high, a low, and some kind of intermediate group. So in the discovery data set, we saw a distinct difference between the groups. We then had 2 independent holdout sets, TCIA data set, and also another unseen Hammersmith data set. And you can see that we have very good discrimination between the low, medium, and high groups. This was both in univariate and multivariate analysis. And multivariate analysis Cox regression analysis adjusted for known clinical prognostic factors was significant in all groups. So the highlights are that patients with a RPV high in the RPV high group had a median overall survival of less than 2 years, greater than 5 years for those in the low group. And the interesting thing is that it outperformed established molecular subtypes, which were derived from transcriptomics data. And this is important as we develop these imaging approach. It's not just another piece of science, it's something that could make a difference clinically in the future. What about the cancer phenotype? What does all this mean by way of cancer biology? Because these are but biophysical measures to which we'll try and assign some understanding. Broadly speaking, we can assign some understanding to these. So FD might represent tumor irregularity, and then we have intermixed stroma with tumor cells, local and global heterogeneity. But we wanted to delve in a little bit more. And so we were lucky to have fresh frozen samples for all these patients. And so managed to run proteomics and targeted sequencing, as I've already elaborated. And what we found by doing a gene set enrichment analysis was that the RPV high group was enriched in stroma, while the RPV low group was enriched in DNA damage response pathway, more accurately, homologous recombination. We created an expression RPV. So this is a set of genes that correlate with the RPV. And we did that because we didn't have enough 3D data across all tumor sets. And so this was a mechanism to explore other tumor types. And we used both ERPV and RPV to mine a lot of our biological molecular data and found that, yes, these were correlated with stromal factors. But interestingly, they did not overlap with cyclin E amplification and BRCA mutation. And this is surprising because ovarian cancer harbors a lot of cyclin E amplification, which is related to outcome. But what we realized by delving a bit into the data was that these phenotypes were non-overlapping phenotypes. So one could combine them in the future to even improve the C index a lot. So this is the example, 2 examples, a clinical trial example using RPV, where we understand the molecular phenotype, we understand the imaging phenotype, and can combine them with the data that we have. So we know that for the high RPV group, they will not respond to the impact of optimal surgery would be minimal, whereas ultra radical surgery would be great in the group with the low RPV. But stromal targeting clinical trials should be encouraged in the high RPV. And we've now locked the RPV, and we're collaborating with various people to optimize that. So I want to talk a bit about immunotherapy. This is very topical in cancer research. This is a cartoon from Science work that Tony Ribas and others published many years ago. PET scanning is being investigated for tumor foreigners, to immune cell infiltration and absence of checkpoints. But here, I think CT in particular can make an impact by looking at the tumor architecture. And a number of research groups have already investigated this. A few of those highlighted here. Work in all solid tumors. We normally start with just a single tumor type, but these guys have looked at all comers in a clinical trial, as well as other studies that have looked at non-small cell lung cancer and head and neck cancer. HCC, hepatocellular carcinoma, and with decent C indices for these groups. Just want to highlight one of these studies where they did a training set, a validation set, and then used the imaging output to test response to checkpoint inhibitors. And as you can see in the lower right panel, there is very strong prediction of outcome. And this is the sort of study that would be recommended, or study design that would be recommended for future supervised approaches. But of course, as we move from the really strict criteria to the real world, the performance drops. And so it's important to appreciate the challenges and figure out what to do. General considerations for challenges would be reliability, harmonization, explainability of features of vectors across tumor types, and within specific lineages as well. In terms of reliability, every single step of the pipeline would have some potential for errors, and I've just indicated a few. And so the acquisition process, the pre-processing, where one might, for instance, look at interpolation methods, but could have errors. Segmentation has huge errors, and feature extraction methods and classification could potentially have huge errors as well. And so a number of organizations around the world have been drawing guidelines. Some of these are very instructive, and I think what I want to say is that these are bad guidelines, and we should try and follow them as best we can, but we should not follow them, with to the extent of stifling the research. So, for instance, it's recommended that one accurately documents, and you can't get a paper through now without describing normalization and resampling methods, or various types of inter-observer or test-retest methods, and or external validity of these. And there is good reason for suggesting these guidelines. But the question remains whether we generate a biomarker and then investigate performance, or we triage all the features and then generate that biomarker. And I think there's room to investigate both options in upcoming studies. This is one example from Chris, who's joined a group as a PhD student, and he's introduced another interesting component which he obtained from Wybra, and that is introducing a null hypothesis. So he randomizes the voxels, and if the vector is a true vector, it should all disappear to zero. And so it's one other mechanism within the resampling phase and feature extraction phase to really introduce some additional components for validity. This is just to highlight the fact that if radiomic features would have inherently different values after resampling, shown from an original image and a couple of resampled images. Another issue which is critical, and in the interest of time, I won't spend too much time on this, but it's critical, is erosion or dilation as a validation to support the notion that if you do change the segmentations, you do degrade or otherwise the vectors that are generated. So the lessons learned to end would be to start with a large data set, to have confidence in the segmentation and have a good plan for validation and testing. And one last slide is what we intend to do as NCITA, which is really beyond our original framework, is to develop these radiomic data sets, large enough data sets so we can test most of the sorts of challenges that I've outlined from multiple institutions and a lot of contribution. So with that, I'll stop, and I'd like to thank a lot of people who have contributed to this, without mentioning too many names, Helnan, Andrea, and various who have really supported this work. Thank you.
Thank you so much, Eric. That was brilliant. I think let's wait for all the three talks, and then we'll open the discussion session, because they kind of flow one into the other, so they complement each other. So now I'm going to share my screen. I'm the second speaker, and my talk will follow on Eric's, but will be more. But it will be sort of more focused on the role of radiomics and habitat imaging, on actually the unraveling of tumor heterogeneity, which Eric sort of touched upon, and that also the group that have done really seminal work. So those are my disclosures. None of them actually related to this work, to this talk at all, but there they are listed. There are several challenges in ovarian cancer imaging, and I think Eric touched in most of them, and highlighted the bottlenecks very nicely. What I'm going to really focus in is the quantification and monitoring of inter-tumor heterogeneity during the course of disease, and how do you really integrate novel tools to do that, and also how do you biologically validate some of those radiomic features, very, very nicely outlined by Eric in his talk. And I'm going to touch upon our efforts that we're doing now to actually integrate these highly annotated data sets of imaging, clinical, molecular data on this patient-by-patient level, with the aim of actually improving the prediction that we do based on various sort of limited clinical variables in the routine clinical practice. Okay, so I'm just going to start off by showing you where the problem lies. And the ones in the audience that are radiologists, I'm sure they're pretty frustrated when they kind of sit and read those cases of treatment response. And these are 3 hypothetical patients. As you can see, I've kind of annotated ovarian lesions here. They come for a CT prior to neoadjuvant chemotherapy, and then after 3 cycles of neoadjuvant chemotherapy, there is another CT. So you're sitting there, looking at the CTs and trying to sort of classify, is the patient has got progressive disease, which is pretty straightforward, because you measure the lesions, you RECIST, and then you know there's definite progressive disease. It's not difficult to also assess or quantify the partial or complete response. Where the problem lies, especially with ovarian cancer, because RECIST is so ill-suited to confirm disease, is actually this big group of stable disease, because some of the lesions might get bigger, some might get smaller. And I know I've annotated only ovarian lesions here, but as you know, ovarian cancer has multiple lesions throughout the abdomen and pelvis. So this group of stable disease actually represents about 33% of the whole patient population. This is really solid data coming from the ICON-8 trial. So in this large group, we actually don't know what to do. So we're putting patients in stable disease, but actually about 14% of them, they actually have good response, and the rest of them actually have disease progression. It's really important to sort of classify this big group in a much better way. And what we want to do, and again, Eric already touched on the whole tumor microenvironment, we want to use the imaging data that quantifies the heterogeneity, not just change in volume, in size and volume, with the tumor immune microenvironment data that comes from the biopsy at the start. So you really immune profile these patients and take this into account, and then you can really make a better assessment if the lesions are really progressing or regressing overall. So this is really the really important to take into account the data integration. So why have I chosen, or have we chosen as a group in Cambridge ovarian cancer is one of the areas. Well, it does actually represent a model of extraordinary heterogeneity. It's a disease that presents as metastatic disease at presentation, not at recurrence, when you have multiple areas of tumors dotted around into not only the pelvis, but also in the peritoneal cavity. We know now in the pelvis, and we know that ovarian cancer originates from the fimbrial end of the fallopian tube, and the first port of metastasis is the ovary. It's not the primary ovarian tumor, it's where the first metastasis land, and then with the current of the site, it obviously gets distributed throughout the abdomen and pelvis. So not only we have heterogeneity between the patients, which we have in any tumor, but also we have heterogeneity between the sites of the tumor as well as within the tumors. There are 3 levels of heterogeneity in ovarian cancer that pose really the challenges. Now we know what the big challenge is, and we know that heterogeneity within a single lesion does not capture the whole tumor microenvironment, which now from the genomic and proteomic studies, we know that drive the resistance in ovarian cancer. And what kills the patient in ovarian cancer, other tumor types, but especially here, it's that resistance to chemotherapy, which unfortunately, ultimately, about 80% of patients will develop, about 50% on the first round, and about 80% later on. But wherever there is a challenge, there is an opportunity, and that's the opportunity to develop the new computational methods to actually map and track this special and temporal heterogeneity in ovarian cancer. And Eric beautifully outlined the methods that their group is using for that. So what we did in our group, and this is work that started at Memorial Sloan Kettering Cancer Center, but then we brought it to Cambridge and sort of changed it and improved it, is to take into account, to develop these metrics who take into account not just inter-lesion heterogeneity, but also intra-lesion heterogeneity. And this is just some example of retrospective work, which obviously doesn't produce the biological validation, but is still important in terms of prediction and prognosis. These are 2 patients with ovarian cancer. They both belong to a genomic subtype called mesenchymal subtype. This is the Clover classification, is the genetic classification, and there are 4 types. The thing to remember is that the mesenchymal subtype is very poor prognosis because it's associated with very high resistance to chemotherapy, as high as 70%. So these patients in general tend to do very badly. Now, even within the same genomic subgroup, you can see here, there are patients which have more homogeneous disease, which survive very well, and the patient which has heterogeneous disease that survives very little, not even a year. Interesting, this is not related to the volume of disease. As you can see in this case, the patient has high volume of disease, although being homogeneous, and this other patient here has low volume of disease. So I think imaging can be quite complementary. Even within that the genomic stratification, we can better substratify patients in terms of their prognosis. And this is in the setting of neoadjuvant chemotherapy, but we've done that. This is work led by Julia Lackman in New York. We've done that in the setting of immunotherapy, and this is in about 70 patients having different regimens and combination of immunotherapy for recurrent ovarian cancer. And the combination of heterogeneity within the largest lesion and the number of sites of disease was a better predictor of the durable clinical benefit, which is what we use for immunotherapy. So how did we actually, how do we move from that vision to more of building really a solid pipeline that we can actually do those studies in a far more robust way, as Eric alluded to in his talk? Well, in Cambridge, we've got this pipeline. We actually start from the raw images. I'm not going to show you any data on raw images yet, but we start from the sinograms, and we move to reconstruction. You can see here bilateral ovarian cancer with peritoneal disease in the pouch of Douglas. And then we've got an image repository, which Simon will go in detail. And this is thanks to Lorena Escudero, which is, I think, is attending, but she's done a fantastic work in actually setting that in Cambridge and making it functional. The idea is that the collaborating center can actually fit imaging here, and then that we can also sort of contribute to their work. So it's all about collaboration. We've now developed a system for automatic segmentation as well as sub-segmentation of ovarian lesions, and I'm going to talk more about that. And of course, we do the radiomics, and again, I'm going to talk more about that. And we have developed our prediction models in terms of treatment response and outcome, and all this leading to obviously personalized treatment. What we feed as well, and we've got some initial work here, is the data integration, which is really crucial, and Eric already beautifully explained how that was done with the network communication paper, when you actually feed the histology, the clinical data, but also what we're including is the circulating tumor markers as the ctDNA. What we're also doing, we're trying to biologically validate our radiomics, and we've developed tools to biopsy those areas which are different, those habitats, being that on MRI or CT, using ultrasound fusion prior to chemotherapy, and then at surgery, we'll develop this 3D mold pipeline that we can actually section the lesions accordingly, so we can biologically validate the surgery. So initially at biopsy, prior to treatment, and we can track those habitats at surgery at the end of treatment. And of course, that all links to prediction again. So there is some really great work we've managed to do with, in collaboration with the Department of Advanced Mathematics and Theoretical Physics in Cambridge. When we actually start from the axis slice, we're using the 2D unit segmentation, but ultimately with using deep learning, we're actually employing a 3D unit for the prediction stack. So, and this is really brilliant work done by Thomas Butenkot at the DUMP in collaboration with Leonardo, one of our postdocs. Really great examples of collaboration across university campus. So in terms of the dice scores we're having for that, we're approaching about 90% in the pelvis. It's a little bit more challenging in the peritoneum, and that is work in progress. But we're getting better and better results up to 0.75 in terms of dice coefficient in the peritoneum. Obviously, the smaller the lesion is, more difficult it is to actually get that perfect segmentation. But I think this is work in the right direction, because you saw how many challenges the segmentation poses, and if we can sort this out, that lays the grounds for really good, robust radiomics work as well down the line. Okay. So this is just again to show you the comparison in the pelvis between the manual automatic segmentation, when actually the algorithm does a pretty good job. So what we're doing is trying to actually fit in a all-in-one that go from sinogram to the segmentation, to the radiomics, to the prediction. And we've got 2 years and funding from Wellcome Trust to do that and integrate it into our clinical flow or prototype. So what Leonardo and Ramona have done brilliantly is actually looked at the sub-tissue segmentation of those lesions, just published in Computers in Biology and Medicine. We know that tumors are heterogeneous. They contain the solid components, the necrotic components, the purely cystic components, sometimes the calcifications, such as in this case here. And we know that the purely solid components are probably the ones that contribute to the circulating tumor marker and also to probably disease recurrence. So it's really important to do this sub-tissue segmentation. The omentum is even more important, because you can actually tease out the normal fat from actually what is really tumor in the omentum. And you can see how the dice coefficients are actually quite high. And again, this is just comparing the algorithm that Leonardo has developed with the others, and showing you the performance actually is one of the highest. Moving to data integration, this is work spearheaded by Mireia Crispino-Truzan and Ramona Wojtek. And this is looking at our neoadjuvant cases on over 100 cases with separate validation set from Apollo program in the US, as well as TCIA, which is linked to TCGA, obviously, as well as the Barts Hospital in London. And this is looking at integrating over time not just the texture features or the volume of the disease, but also the treatment type, because obviously it's going to change depending on the time of the treatment, also the time where the treatment, some patients get 3 cycles, some get 4 cycles of chemotherapy. And this is all important if you want to really optimize your prediction at the end, your model for prediction. And as you can see here, along these points of the CT scan and the chemotherapy session on these rounded sort of circles, you can also see the other information we're collecting, which is the sites, or not collected, but integrating through diffusion, the number of disease sites, which are more semantic, as well as the total volume and the ctDNA. We're using the team out here. So, our philosophy is integrate semantic, clinical, radiomics data with a ctDNA, and this is just the early result. And this, as you can see, this is just a model development set. And our model already does much better in separating the patients which do well and those who don't in terms of progression-free survival, compared to just a simple model that is now used in the clinic, which just take into account the age and the state of disease. And this is really exciting work. And this is the sort of thing we want to share the expertise, the images through the NCITA, and combine with the other centers, so we can exchange not just our images and the segmentation, but also the various algorithms developed in different sites. Moving on to some proteomic works, and this is really elegant work done by Lucien Beer in my group. This is a really great collaboration with the NCI, with the TCI, TCGA. So what we did here, which was very elegant and this is all thanks to our statistician at the NIH, at the NCI, was the way we prioritized the proteins which were candidates in the ovarian cancer patients, because as you can imagine, there will be hundreds of proteins and hundreds of features, and you've got yourself intermediate intermediate comparisons. So what we did, we looked at we had 169 patients on the TCGA and the CPTAC, which is a proteomic database. And, the way we reduced those proteins was actually quite elegant. At the end, we ended up only 16 proteins which were involved in the amino acid metabolism. We did this by looking at the overlap with a single case which had multiple biopsies at multiple sites. And what we looked at was overlap with this highly annotated case with the whole database of the CPTAC. And you can see also how we reduce the features that we use in terms of the heterogeneity. We only use 3 CT-based features, which take into account both intra and inter site tumor heterogeneity. And the numbers are small, but also the features and the proteins are reduced. However, this is just a hypothesis-generating study, and you can see several associations here. This is allowing for multiple comparisons, after the Bonferroni test. And you can see the disease in a mesentery, which is associated with a poor prognosis, associated with one of the CRIP protein, which is now actually being confirmed in our larger data set, which is work in progress, that is spearheaded by Lorena, Leonardo, and Lucien. And you can see other associations here, both with texture as well as the imaging traits. So moving into the heterogeneity itself and how do you biologically validate it? Obviously, when one does the biopsy with the ultrasound or the CT, you'd go for just a site which is safe in the disease. And of course, even when you do the radiomics directed biopsy, you go for safety first, but you can actually do much more. So by actually mapping this radiomic on top of the CT and finding new ways to target these biopsies, really important, because the hypothesis is, as Eric already alluded to, that these areas would be different in terms of the underlying histology and also underlying genomics as well. And can you actually, in a way, infer from the CT rather than making those patients go through multiple biopsies, which is not practical, really. And what the group has done here, and this is led by Paula Martin Gonzalez, is using the principal component analysis to actually derive those habitats, and we can then target using our new tools. And our new tool, which is really a baby tool, because we've only worked on this for the last 16 months, but the last 6 months, actually minus the last 3 months we've been in lockdown in a way. But what we wanted to do is develop a tool that you can actually accurately sample those various habitats to actually understand the underlying biology in the neoadjuvant setting. So what we've done so far is actually setting the framework, which works, and we've integrated it now in the clinical work. Obviously, some of it has been paused now due to COVID, but now we can do this with only adding about 10 minutes to the biopsy. So it's the same session, rather than just doing the simple biopsy, we overlay the radiomic map with the CT, and then with the fusion ultrasound. I'm going to show an example. And all goes sort of relatively smoothly. Obviously, some preparatory work to do the segmentation, sub-segmentation, texture mapping, extraction, clustering, and then a DICOM export, and then everything is ready in the machine, all transferred for the person who does the biopsy to do that. In that case, Helen Atley, one of my wonderful colleagues, is doing those. And of course, we obtain clinical samples and the research samples. Just an illustration here as well, so you can see the tumor outline, the computation of the map, and then at ultrasound, you fuse those, and then we actually target those areas. Now, movies usually don't work. Let's just see. Oh, it does. So you can see the needle going in and sampling the green habitat here, and then there will be another pass that actually samples the red habitat. So we're not yet perfect on that. We're working with kind of medical system to actually adapt one of the systems to correct for some of the motion. And we've got now a new electromagnetic tracing device, a tracking device for the position in the body, which will give us the coordinates to actually improve that. So that will be the sort of next reiteration of the method. But how about the information that MRI could give, which actually it's more, it's in a way physiological and metabolic information compared to CT, which wouldn't contain that. It will really be enhancement in a way. So this is a study we did a while ago at Memorial Sloan Kettering Cancer Center, and this is looking at diffusion. So F from IVIM and D, which is the ADC, but calculated from the IVIM, so the high B values. The K-trans from the perfusion and the FDG PET. Those are co-registered on a voxel by voxel basis with each other. And then K-mean clustering algorithm is run to actually derive those habitats, which means that each habitat is a unique combination of diffusion, perfusion, and metabolism. And in each, and that will be a patient a specific habitat rather than across patients. We've developed another methods for across patient, and I'm going to touch briefly upon. So then these are outlined on MRI. The mold is printed accordingly. Every single opening in this mold here where the knife goes actually corresponds to an image on MRI. So you can really go through and cut that piece, which corresponds to the slice in MRI. And of course, I've only showed a slice here, but if you go up and down MRI, the green and the red and the blue and the yellow will extend through multiple slices. So you choose really the slice in the middle to allow for some movement of the tissue. However, we find that for ovarian cancer, the tissue is actually quite rigid, so it fits quite well in the mold. For the omentum, can be a little bit more tricky. It can disintegrate at times. So then samples are taken, and then half is sent for histopathology and half for genomic analysis. And what our cases showed, and this is just one example of them, is that, of course, all these patients share a T53 mutation as the trunk of the tree, because they're all ovarian cancer. If they didn't, we would query if the pathology was correct or not. And you can see evolutionary, genomically, the ovary blue habitat is not just at imaging, but also genomically, it's on the same evolutionary branch as the omentum blue, which is the metastatic part, and the ovary green and yellow kind of branch together. What is interesting in the blue habitats, the mutations which were subclonal in the ovary blue became clonal in the omentum blue, showing that the disease became more genomically heterogeneous as it spread along the peritoneal cavity. Again, remember, ovary is the first port of metastasis, and then it goes along in the omentum. So the other thing we did, we also looked at the tumor microenvironment by using the multiplex IHC on those lesions. This is another case just to show how different those are: the pouch of Douglas disease, the omentum disease, as well as the ovary here. And this is another mold. You can see here this corresponds to the omentum, and this to the pelvic disease, and there will be another one printed for the pouch of Douglas. And this is the group picking up the samples from the theater, which are wonderful research assistant Ramon coordinated everything. What we found here is that within the lesion, not just between the lesion, but within the lesion, when this is the samples targeted by those habitats, you had the immune hot and cold microenvironment. In the immune cold microenvironment, you had increased mixed signaling and Wnt signaling pathway, and several parameters as outlined here that are associated with several pathways here. So this is just on 10 patients, it's preliminary data. However, it's really strongly suggests that immune heterogeneity in the treatment-naive ovarian cancer is important, but also following treatment, and for that we only have CT data, that neoadjuvant chemotherapy can actually cause immune system activation. And this is important because those patients will do well if you combine with the immunotherapy, not just at recurrence, but actually upfront treatment. This is what we're planning in Cambridge to actually design this new study to move this combination upfront rather than recurrence. So I think, you know, to finish it off, what I was trying to convey is sort of a system for real time tracking of tumor dynamics, not just in clinical trials, but you also saw in clinical trials first and then into the clinical practice, which will start with the segmentation. I've shared with you our work on segmentation, habitat definition being that CT or MRI, obviously they are quite different, the way you define them, how you can do the in vivo targeted sampling using ultrasound, fusion, and of course the 3D tumor mold at the resection. And ultimately, one has to be able to integrate all this data when we develop machine learning models to actually do that and predict the outcome. This was the end of my talk. I'm very grateful to all the collaborators from both sides of the ponds and everywhere, and to our really fabulous group we have in integrative cancer medicine, which is actually making all this work possible. Thank you. Right, I think now we're going to move to the second. I'm going to now Simon Doran, which is going to give the third talk, the NCITA image repository. And Simon is basically ICR and has been absolutely instrumental in getting this going. So Simon.
Hi, Evis. Can you hear me okay?
I can.
Good, excellent. So many thanks to Evis and also to James O'Connor, Amanda, and the rest of the organising team for inviting me. So I am, hopefully, if this slide will move forward. Okay, so I'm going to be talking today about a journey. It's a journey that started many years ago. For me, it was when I moved to ICR in 2006 with an informatics brief. But for the NCITA partners, the collaborations began probably longer ago than many of us care to remember. And they've matured throughout the period while we were funded as Cancer Research UK cancer imaging centres. So this is a journey that starts with a vision that I'm going to talk about today. And to achieve that vision, we had to assemble a team and also to have a plan. So I'll give you a brief rundown of what our plan is for NCITA, but it's much more instructive, I think, to talk about the achievements so far. So even in the short 6 months that we've been in place together, there's a lot to talk about. And it'll be much easier to show you by example what we've done. So this goal of big data nirvana, if you like, is still many years in the distance and by definition probably isn't achievable. But nevertheless, I hope that what I'll show you in this talk is and convince you is that we're headed in a good direction. So the overarching purpose of the image repository is to improve the quality of our science. What I've shared with you in this sentence at the top is the opening sentence of the NCITA case for support. And I've highlighted a few words that might be useful. So let's start with efficient. Even before the current lockdown came into force, our clinicians were already realizing quite how liberating it was to be able to work with their research data anywhere. So if you're at a conference, you need access to your research data. well, let's log on to XNAT and get it. But since March, this has become essential, and the XNAT image repository that I manage and the tools that we've created are enabling PhD students, clinical fellows, and all of us to continue working. So thanks to what the repository's achieved, we can visualize, annotate our data in ways that are becoming ever more like an online PACS. We're aiming to make our data fair, that is, findable, accessible, interoperable, and reusable. We've got a lot more to do with procedures, but the whole idea of NCITA is that by incorporating robust procedures, we can improve our image governance. By integration with the NCITA QA, QC, and trials units, we're going to be in a position to open trials more easily and manage them better. And so what we're hoping as the NCITA repository is that we're going to act as some sort of beacon, an example to the community of how we can handle cancer imaging data well. But more than that, we want to focus on this cross-institutional thing as well. And we want to demonstrate that cross-institutional systems allow us genuinely to create a synergy between our various expertise, not only to achieve more in a given time, so just increase the volume of research, but because we can do things faster and better with our new tools, we can actually achieve things that couldn't be done before. And I'm sure, moreover, everybody knows about this whole repeatability crisis that people are talking about in cancer science. And what we're aiming to do is make all our analysis repeatable and auditable by giving all our data a proper provenance. So before I go any further, I thought it's probably worth pointing out a couple of things that the repository isn't. So I don't know what you might understand by the term repository, but maybe you just think it's a data store. And you might think that what we're hoping to provide is just the sort of equivalent of the image shopping trolley of Amazon. But that would vastly underestimate the potential of what we can do. Yes, we can deliver you images, but we can also do data processing, image visualization, and a whole host of other things. We can execute complex queries. So maybe find me all the patients with a lesion larger than a centimeter in the left femur that were annotated by Professor X between January and April, and who've also had 2 rounds of chemotherapy. One of the big aims, therefore, is to provide for data integration and build cohorts that you just couldn't assemble in one imaging center. But equally, though, even though we've got all these sophisticated image processing and annotation tools, the repository isn't aiming to duplicate the functions of a hospital PACS. A PACS is great for the clinic, it's perfect, it's optimized for routine use, and you can do things with a single click in order to minimize your time as radiologists. But here's the rub. Because everything has to be very strongly CE marked and all that sort of stuff, new features appear only infrequently. What our aim is to be agile and to allow end users themselves to be able to install new tools, new methods. So we want to make it easy to browse, load, and manipulate all types of imaging. Thanks to XNAT, which is our central imaging archive platform, we can do this for DICOM data straight away in a way that's open, vendor neutral, and extensible. But what about non-DICOM modalities, things that you don't see on a PACS? Out of the box, XNAT will allow us to upload and store data files of any type right together with the DICOM data on the repository. But it needs to be taught how to use all the metadata that come along with them. So we've already established the principles in prior work, and the task is to step through all these additional data types and do the coding. You might wonder a bit why I'm showing some preclinical sort of modalities here, but it's worth bearing in mind that some of these things that right now seem quite a way off routine clinical use and routine use in trials are going to come on board at some stage. Siemens is already offering MR elastography as an extension on almost all of their standard platforms. And early phase trials of photoacoustic imaging, for example, are already ongoing at NCITA institutions. So our paradigm is to make all our analysis data aware. So data are pulled off some canonical data set, and the results of that processing are stored back into the database alongside those data, together, crucially, as I said before, with their provenance, allowing us to go back and audit all our studies. Finally, we want to be able to use any search term that we like in the image metadata, and eventually we'd like to go further and to be able to use machine learning to perform image recognition and content-based image searching without humans having to do this laborious tagging of images. And then all of these resources, including the enhanced visualization that goes with them, will be brought together to allow clinicians and scientists to participate in virtual tumor boards. Again, taking advantage of the web-enabled aspects of this so that we can take advantage of expertise wherever in the world it happens to be located. Now, not only do we want to see access to all different types of imaging, we want to be able to access data at all the different levels that you might see in the hierarchy, all the way from the results of whole trials and sets of trials, drilling back down through the levels of the hierarchy, down to the level of a single image voxel if necessary. So why is that important? Well, let's think about the data set from the video that I had on the previous slide, which is courtesy of Matt Blackledge at the ICR. And what we're doing in this slide, in the graph at the bottom, is taking each individual metastasis and plotting a histogram of the ADC values. Now, all of the histograms are different, but previous experience tells us that their shape and the value of ADC at the peak can help us distinguish between different types of lesion. If we then choose to analyze not just the statistical features of the data, but also other properties, for example, texture, maybe add some clinical data in and some genomics, and then we have the type of radiomics prediction that Evis and Eric have just been talking about. If we step up another level of the hierarchy there and look at the whole patient and see how this is distributed, then we get to the stage where maybe we can make a prognosis for what's going to happen to the patient. And you can see here some nice work from Matt Blackledge and co-workers at the ICR, where there's a very neat correlation between survival and the total volume of lesion across the whole tumor volume. If we go another step in the hierarchy and look at a single patient at all time points, we can see that quantitative analysis of the type that we've just talked about shows that there are two distinct classes at the start of treatment. The red lesions have a low average ADC, while the green ones are higher. This is a 49-year-old, if my photo's obscuring the text, it's a 49-year-old woman with ovarian cancer and 4 large masses. So if we step through the various imaging visits, so there are 10 visits altogether, we could actually plot a graph for each lesion across the 10 visits. If we step through those, we see that an interesting thing happens. One set of lesions has an increase in diffusion coefficient and the other has a decrease. And we can use the image repository to marshal all these data in a very easy fashion in order to draw all these data together. So finally, if we step through the different levels, we can see that stages 8 and 9, we can look at efficacy. And then when we come to the stage of all trials, think about what you could do. If you could take all the data from all the trials, put them on the same database, then you can imagine how much easier it would be to perform meta-analysis of the studies and audit the entire process. So here's my version of the flowchart you saw in Evis's lecture just a few minutes ago. The feature of the repository that I want to emphasize here is how ubiquitous it's going to be. In the figure that you can see, the blue and grey, or blue and silver versions of the icon refer to, they mark the places where the repository is doing pretty much what you might traditionally expect a data store to do. That is, to facilitate the transit of data from one system to another. However, the icons in red represent something that's potentially much more interesting. One of the phrases you might remember from earlier is that NCITA aims to create pipelines to produce quality assured, locked down biomarkers. Well, once we've locked them down, what we really should do is then shrink wrap them, and it makes sense to employ them systematically. So one of the attractive features of the XNAT platform is that we can insert what we call triggers, such that when a data set reaches a particular phase in the pipeline, we can run an appropriate set of processing code automatically. So imagine how this could speed up research and speed up discovery. For example, look at the annotation section. As soon as a radiologist has drawn an ROI, ran a tumor, and pressed save, then we can automatically execute our radiomics generation procedure, rerun the modeling code, and get an almost real-time update of the difference that this individual patient made to our AUC. Finally, the icons represented by the yellowy green color represent entirely new functions that we're enabling with the repository, where the repository is interacting directly with users, either to input annotations in the form of an electronic case report form, or to visualize data. And this is just a quick shout out to demonstrate that all of this vision is not just pie in the sky, but it's something that's in process right now. This is one of our sister projects, the CRUK Sarcoma Accelerator, which has just started. We're going to be combining clinical data, digital path, radiology, and multi-omics, all with XNAT acting as data integrator, data aggregator, and it's going to be the first point of contact for potential end users. And the last thing I want to say about our vision is the thing that makes NCITA as a whole unique. This effort is going to be successful because of the tight infrastructure integration between the three different pillars of NCITA, are the trials unit, the QA/QC unit, and the repository. We've got coherent aims, coherent procedures, our data will be joined up, our hardware is going to be co-located, and we have weekly managers between the unit managers, weekly meetings between the unit managers to ensure that trials are joined up. So you've seen our aspirations. Now here's the team that's already delivering this. Avis is the PI and chair of the repository unit, and she's got Lorena and another Avis staff in Cambridge. I work with James Darcy at ICR, and then we've got Mo Al-Sad, Cristiano Torti, Paul Tarr, the other institute, and Chris Rookyard at King's. So in fact, next Wednesday, we're recruiting for a final position in the repository team, who's going to be a senior research radiographer based at UCL. The practical implementation of this vision divides fairly neatly into two parts: the bits that the end user sees, and then all the hard work that goes into the invisible infrastructure that ultimately delivers the service. Like the software on your phone, you should be able to expect that this just works. Our aim is to make the connections and the data flow as seamless as possible, and the better we do, the less you'll notice us doing it. And our aim is to hide the complexities of what's actually happening. So let's look at first at the things that we hope to do to make your lives as researchers on imaging trials easier. We want to start with the thing that we think is going to be have the most impact, and it's going to be most visual and most noticeable. So as Lorraine has already found in Cambridge, being able to view and annotate the research data is already proving extremely popular. Next, we want to exploit the unique advantages of the NCITA network, and I'll talk more about XNAT Federation in a later slide. I've already discussed the standardized data curation and processing algorithms and fair data access. But one of the major things that we're obviously going to be doing is accumulating data. And Chris Brew-Graves, the NCITA project manager, has a list that's getting ever longer of studies that want to be adopted by NCITA. So we hope to have no shortage of data coming into the platform. And all of this is going to be delivered with a tight focus on the physical, technical, and procedural infrastructure. And of course, ultimately, none of this is going to be any use at all if we can't make this sustainable. If we accumulate data for 5 years and then the NCITA grant vanishes with all the repository, then that would be no good. So we're working hard and already have the start of mechanisms to create this as a sustainable service. So I've talked a lot about our new image viewer. So this is what it looks like. The initial integration work was the product of a really fruitful collaboration between James Darcy on the left and James Petts, who was another software engineer who worked for me last year. Now, unfortunately, James Petts, the bad news is he was so good that industry spotted him and made him an offer that he couldn't refuse. The good news, however, is that Radical Imaging, the company that poached him from me, is a key member of what's called the Open Health Imaging Foundation, or OHIF for short. And this brings me to a really important take-home message. What we're delivering here is open source infrastructure. Now, this means not only that it supports the interoperability aspects of our FAIR data agenda, but also that we benefit from the fruits of other people's labor as much as they gain things from us. So this collaboration effort really isn't a zero-sum game. Other people take our code, they develop it as part of their projects, and then they contribute back amazing things to us. So the microscopy viewer that you can see inset in there is a project that's already available on the OHIF in embryonic form, and James Darcy is going to be spending time crafting it so that it works within XNAT. So it's worth pointing out that we provide, so the NCITA consortium provides the official release for the XNAT the viewer on the XNAT platform. Our latest release was in January 2020. It's already been downloaded by more than 500 people, and there's evidence by citation that it's already being used actively in real and relevant projects. So collaboration is a key element of what we'll do as well. So what you're seeing in the video that hopefully is just playing now is an initial proof of principle of the integration of NVIDIA's Clara AI-assisted annotation platform. Obviously, the ultimate motivation to NVIDIA for helping us is so that they can sell more GPUs and more hardware. But the way they've gone about helping academics is both imaginative and exciting. So what they do is they provide for free Clara, which is an SDK, or software development toolkit, that allows academics to develop their own segmentation models and integrate them within a framework. Now, this is going to be absolutely ideal for all those models that Evis has just talked about. What we're going to be able to do, and Lorena, I suspect, in Cambridge is going to be helping us do this, is to take those models, incorporate them in the viewer, and this provides a route for getting the models from academic research to clinical trials in the shortest possible time. So now, OHIF also contains a whole bunch of really flexible annotation tools, and you can see what's there already. The red one and the green one are ones that we as NCITA have introduced, so the contour-based and mask-based segmentations. And crucially, these are the only ones at the moment that can be captured in a machine readable form. So what you can do at the moment is just, you can annotate and add text to your heart's content, but all you can do is capture that as a JPEG or something like that, that can't contribute further to the research process. Moreover, there aren't really any good interoperable annotation solutions in radiology generally. The current state of the art is RSNA's RAD report template in the MRRT format, but that's very limited in the types of information it can store. So what we've done already is to get with the state of the art by implementing RAD reports in a modern web framework that we could put into the viewer, but also Mo's program for the next little while, once he's finished doing the AI segmentation, is to capture all the rest of the features that we can annotate and output them directly to machine readable DICOM structured reports that you could take into any other machine learning application. Now, the problem statement for this next application is really easy. Okay, what I want to be able to do is go in, is take the green region of interest. I want to press Control-C to copy it, and then move to a different partition on my display, press Control-V, and have that region of interest copy over. Sounds really easy, right? But it turns out that enabling cut and paste of regions of interest is a really non-trivial task. So different fields of view and regions of interest, or different fields of view and different matrix sizes within the same imaging session create problems. Patients may move. There may potentially be differing spatial distortions between MR sequences, even within a session. And then when you go between modalities, so let's suppose we wanted to annotate on MR and paste it onto a CT image, or paste it onto the PET or whatever, those different contrast mechanisms between modalities make it problematical to do registrations sometimes. So Cristiano's just starting out on this. We've already had some successes. In the bottom, you can see an image region of interest with the source region. The target image is clearly shifted in the slice direction and rotated with respect to the source. So you can't just do a straight cut and paste. But on the right is the region of interest correctly registered and pasted onto the target image. Chris Rukyard is our man for looking at XNAT federation. So there is a previous example of a federation between XNATs in the UK. It's called the Dementias Platform UK. But I want you to notice a key difference between the architecture of that collaboration and the way we've set things up. Our aim is slightly different. Rather than have a whole set of sites that publish to some master version of XNAT and create single locked down, frozen exemplar data sets, our aim here is much more agile. We want researchers to search among a genuine federation of peers at different sites. Sites need to be able to keep jurisdiction over their own data. And crucially, everybody who logs onto XNAT will have their own profile of data that they're allowed to access. So a search under our new XNAT federation query will give you different values depending on different returns, depending on what consortia you're hooked into, what agreements you've got PI to PI, and so on. So I won't talk very much about infrastructure, although there's a lot of work here. Just to say that at ICR, we've been working for many years on this. We have more than 170 separate XNAT projects. And we've already demonstrated the ability to facilitate multi-center clinical trials. So HALT is a radiotherapy trial that has 26 open sites already, with the data all being managed on XNAT. Cambridge already had an NCITA, Cambridge already, prior to NCITA, had an XNAT in the preclinical sphere, and various different departments had their own. But now, since Lorena Escudero has started, there's been a new NCITA XNAT instance. It already, since about January, has 8 projects with almost 1,000 imaging sessions uploaded already, including a major contribution to the COVID effort. Mo has set up a clinical XNAT at Imperial as of now. Paul Tarr has had the existing Cancer Imaging Centre XNAT at Manchester transferred to us, and is about to relaunch it this summer. And finally, at KCL and UCL, there's already significant expertise in XNAT, and there are discussions ongoing about just how best we can interface all this together. So I just want one quick word about collaboration between other units. So we've already talked about the robust biomarkers. How are we actually going to deliver this? Well, XNAT has a sophisticated, what's called container service, based on widely used Docker framework. I don't have time to explain that to those of you who aren't familiar with containers, but just to say that what it'll eventually mean is that we developed, as I said before, these shrink-wrapped containers that contain our standardized procedures. And once we have them, we can execute these with a single click on entire data sets and patient cohorts. So I'm sure you can imagine how much easier that makes it to analyze data. Suppose you do analyze the whole data for your trial, then realize that you want to investigate what happens if you change a single assumption and change a single algorithm. Well, you can re-implement that as your Docker container, upload the new container to the server, press the button again, and it'll do all the analysis in only the length of time it takes to achieve the computation. The work that we've done so far, we've already got places. So Matt Orton in the QA/QC unit has been working on the interpolation to standard formats of images. James Darcy and I have been collaborating on radiomic feature generation using an in-house MATLAB code. And Matt Orton has been working on a comparator, so we can have a direct comparison between 2 supposedly equivalent sets of radiomic features using PyRadiomics. And finally, Paul Tarr in Manchester is working on incorporation of some QA/QC software called Tina Vision. Okay, so this is my last slide, and I thought it was worth trying to assess where we are on this road to big data nirvana. So here's another way to look at the earlier graphic. Without a genuine vision, there are just too many avenues to explore and not enough cash to explore them. The world of big data is very big, and so we need a direction, otherwise we flounder. Once we knew where we were aiming for, the next step was to try and convince people that our ideas had some potential. I think we've done that. We've produced a prototype. We've demonstrated that it does something useful, and we've now moved a long way towards the case where we're already opening multi-center trials. With the investment that NCITA's brought, we finally get to the stage of reaching a wider audience and delivering some real science. So Nirvana's still a long way off, and probably, as I said, by definition, we probably never get there. But the next few years are going to be really exciting ones, and what we do now is going to be crucial for the success of turning that vision into a reality. So thanks very much, and hope you'll ask me any questions that you'd like.
Thank you so much, Simon. This has just been wonderful. Before I open to the audience, thank you to both you and Eric for your excellent talks. And I think everything flowed, and we covered a lot of areas within the limited time. But now it's open to questions.
Hi. Hey, Evis, this is Tarek from MD Anderson.
Hi.
How are you?
I'm
Good to
fine.
hear.
Oh, that was early for you. Thank you for joining.
That is true. It's very early. I couldn't resist. I couldn't resist myself, joining. It was excellent talk. Thanks for really giving a good view. I think I heard part of it at SAR in March, and I think it's very informative. I have a question for Dr. Doran or Dr. Simon. I'm sorry if I...
Hi, yeah, I'm Simon.
Hi. I'm an abdominal radiologist, and I'm actually interested in getting XNAT into our system. So how do we get started, and what are the resources we need to align to actually have this repository? I heard that in the University of Washington had some kind of a pilots or initial studies done. I heard through some of my colleagues at Akron the last fall meeting. So just want to hear from you, from your experience.
Certainly. So I think I guess you've already navigated to sort of the xnat.org website. So there are two things to say. The first is the group at WashU, the core site for XNAT, and Professor Dan Marcus, with whom we have a very good collaboration at WashU, will, I'm sure, be delighted to see new people. They have a lot of information on their site as regards setting up XNAT. But one of the interesting things about NCITA is that, as I said, we're trying to be an example of really good practice in the field. So we have an NCITA website up and running, which actually has technical details of exactly what you need to do to set up an XNAT. In fact, because we're currently in the process of setting it up in a coherent fashion at least seven different centers, we actually have 3 separate examples of how people have set it up at the different NCITA centers. So you can actually sort of customize your setup. There are 3, or probably more than 3 different ways you can do it. And you need to tailor that to the specific resources that you have in your local consortium. So do contact me afterwards, and I'll be happy to point you in the direction of all these instructions.
Excellent. Thank you.
You can either unmute and ask your question, or you can put on the chat box. Okay, perfect. Actually, Lorena answered the question. We have info in our NCITA GitHub repository. Question for Simon. Really interesting talk. Thank you. What staff resources do you need to build up and maintain the repository? KCL.
Okay, so well, I guess the answer is you've seen the team that we have. We're currently setting up and maintaining XNAT over, I guess, 7 different centers. A lot of the staff effort initially has been in doing the setup. So Evis will confirm how hard Lorena has worked to set this up. So crucially, it's not just technical. So that there's a technical piece, there's a procedural piece. So one of the biggest headaches is, how do I get the images from, how do I persuade my institution to let me send my images into the repository? So that varies from institution to institution. Then there's an education piece, so explaining to users how exactly they should use it. And then, so that's that's the setup. And then, of course, the really exciting bit is how you develop new tools to go on it. And that's where you need additional staff with programming capabilities and skills to deliver that. So really, the world's your oyster. It's what do you want to deliver? It's completely customizable to you to deliver it the way you want.
And I think once you have that system in place, you'd see interest from other groups to actually use it and kind of create more collaboration. We've seen it with the COVID-19 that we are hosting in Cambridge. Then it's a lot of excitement around the NCITA. And you might say, well, this is not cancer. Well, we're following up those patients. We have all the images. So, it's a very rich, imaging source and other data source. So. Okay, any other questions? Yes.
Hi, Evis, I have a question.
Yeah. Oh, okay, Roland.
Hi. Hi. I'd just like to say thanks to everyone speaking. It's been really interesting and to hear what everyone's doing. My question is really about image acquisition and whether there's going to be any move across the NCITA group to standardize image protocols, actually the way that you acquire the imaging, because obviously it's fine about post-processing, but you have to know what's coming in the front door. And as we all know, radiologists like doing different things. Is there any way, is there any move to try and get some kind of unification of practice, or is that a step too far?
No, I think but Eric actually chairs the QA QC, so Eric will be in a better position. I can follow up because I think it's great to standardize, but the world is not standardized, so we have to attack it on both sides. But I'll let Eric fix that.
So I guess the starting point is that it's not standardized. We would like to shift towards the standardization of several of our processes. And what we are doing right now, and this is led by Métis Jariville, who is leading the QA/QC group, and she is beginning to implement a quality system. And I think once that quality system is set up, then we can all work to setting qualities. I think initially it will be quite a challenge because several trials are at different stages. But for newer trials, I think the direction of travel will be some kind of standardization as they go through the NCITA trials unit and as they go through NCITA's quality control, quality assurance unit.
The other thing we've been doing, and actually Lorena in Cambridge is doing a lot of work on that already, it's actually, the data is what it is for some of the big retrospective studies that you really want to mine in terms of some of the radiomics. When you talk about CT, you can never standardize CTs across multinational, multi-center studies. But what we're doing is, what are the post-processing steps we can take in account to actually make them as robust as we can? So that's another step. So we're kind of attacking it for both sides. I think there are questions on the chat box, and I think maybe most of it actually is for you, Simon. I know. Is it possible to upload the data? This is Lucy. Is it possible to upload the data to the repository from a site that doesn't have its own XNAT server?
So, short answer, yes.
We want to.
Is it possible to anonymize on upload is a more interesting question, I think. And the answer is yes, it's entirely technically feasible to anonymize automatically on upload, but you probably don't want to do that because it probably breaks any institutional agreement or that you have in running the trial. So we can provide tools that, in fact, one of the quality assurance steps, and one of the things that will be on our documentation server, is precisely details of how best to do that anonymization locally. So you should probably never let data leave your site anonymized, although it has to be said that what I often run is an automatic check. For example, remove private tags automatically for anything that comes through onto certain repositories, because you just want to make absolutely sure that you're not importing anything that's got any metadata that you don't want. So in short answer, yes, it's possible to upload, easy peasy.
And then I think it's a follow, there's another question. If you allow the researchers to add their analysis in the repository, how do you manage the quality of the analysis data and the assurance everyone else who comes across it? If other groups want to use the NCITA, is there a charge for it? Depends what you want to use it, but Simon, you're managing that. I don't want to put words in.
So first off, if I allow researchers to add their analysis to the repository, so I guess the first point is, if you control your own XNAT, then you can do whatever you like, and it's caveat emptor. But if a user wants to install their methodology on a system that we run, I think idea is for lockdown procedures. So that methodology would go through the NCITA QA/QC unit to be verified first. And then once that unit was happy with it, they would talk to me and I would say, right, shrink wrap it into a container and now I'll install it. But there'd be an NCITA process for allowing people to do this, I think.
Perfect. And I think it was a follow-up question on Roland, and I think probably the answer is yes. Would the unification of scanning techniques be something the UCL radiographer would be involved in?
I wouldn't be so. I would strongly expect that that would help. The idea, I think, will be that such a person would travel between sites and help standardize procedures. Exactly.
Excellent. I don't think there are any more questions in the chat box, but any questions not in the chat box? No. Okay, well, you know, everybody could do with a little bit extra time that you didn't think you had in your day, you know, from one Zoom call to another, maybe sort of doing a walkabout or something like that. Well, I think I want to thank you, the speakers, thank James O'Connor for actually organizing the whole event, and the participants who actually makes it even more special. And yeah, and then stay safe, and don't forget the next webinar, which I actually even forgot the date when it is. I think it's the 26th, right? 26th of June is the next webinar, I was right. Perfect. See you all.
Thank you.
Thank you.
Bye.